Good morning, everybody, and welcome back to BioSC 140. This video is group problems. So it's kind of a review-ish type video, preparation type video for exam one. First off, a few things about exam one. It's worth 100 points. It covers lecture material. If you saw something in lab, but you did not see it in lecture, it's not on the test. If you saw it in lecture, but not in lab, it's on the test. If you saw it in lecture and lab, there's a very, very good chance it's going to be on the test because it's so important that I put it in lecture and lab materials. So it's over lecture materials. If it's in lecture material, it's on the quiz, on the exam. Materials to study. I would focus my efforts on the lecture slides, or what I would do is when you're reviewing, I would have your lecture slides up. I would have your handwritten notes that you took while watching the videos, and I would have the class notes all out in front of me at the same time. I would go through the slides as I go through my handwritten notes and the provided notes. And I would just go slide to slide, making sure I touched on everything. I'd pay special attention to big ideas and any vocab word. It's easy to write test questions on vocab words. It's really easy. So if you see a word that I repeat, if you see a word that is bolded and underlined, a key term, learn it. It could be in a question, it could be in the answer. And just go through all the slides and all your notes, seeing each topic, seeing each thing in the 3 different forms. Take your time, read a slide, read the notes for the slide, think about it, roll it around in your mind, process it, and move on. After I feel comfortable with that, I would consult the group problems. You need to consult the group problems. This is my 8th semester, been doing this a while. Students who only look at the slides tend to not do as well as students who look at the slides and the group problems. Typically, what we do with the group problems is we get into groups. I know, shocking. But we break the class up into 6 groups. And I assign each group one of the word problems from our group problems. That group then makes a presentation for that problem. And then we have group 1 come up and teach their presentation for question 1 to the whole class. Group 2 teach their presentation for question 2 to the whole class. And it's a really good way to review, a good way to study in small groups and just prepare for the exam. Now, we're online, so that makes it a little bit tougher. We're not able to do that. But it's still good study material. It helps you see, so group problems are often pretty applied. It's the knowledge that we've gone over kind of applied to a situation often. And it really helps solidify your knowledge at the level that you need it to be. Quite often, students have a level of knowledge after watching the slides and they're going over the notes. And then group problems just kind of takes it to that next level of understanding. So make sure you go over group problems. My recommendation is you should be at the point before you take the exam where you can answer all of the group problems without any help. Like, I give you the group problem and you can write down a complete answer with no help all by yourself. That's the level you should be at before you start the exam. So let's get into it. Outline the 4 basic themes of physiology and give specific examples of each. You should be able to do this. You should be able to, recite the four main themes of physiology and give examples for each. So let's look at this. Structure and function. Structure and function is important because it was like one of the first things we talked about the whole semester. What can you think of some examples of structure and function right now that we've talked about? Well, we talked about the rabbit's ears, about how the rabbit's ears allows for thermal regulation. We talked about how the knees on the x-ray, how when there's arthritis, when there's degeneration in knees, you can see how the structure changes and therefore the function changes. Molecular interactions. We've talked about induced fit and locking key model for enzyme function, receptor function. Size and shape, form and function go hand in hand even at the molecular level. Mechanical properties, so think of the knees on the x-ray. And we've also even talked about compartmentalization, how we have these tissue membranes and cell membranes and membranes of organelles that allow for different compartments within our body. That's an example of form and function. Biological energy use. We've talked some about this so far. We will talk a lot more about it for test 2. Some examples, well, how about muscle use? We've talked about muscles. That's an energy using process. We've talked about active transport. Active transport, by definition, biological energy use. Communication. So cell-to-cell communication. How many cells do you have in your body? A lot. And they need to work as a coordinated team in order to have a successful human being. Well, we've talked a lot about communication. We've talked about hormones, insulin, glucagon. We've talked about the nervous system, and we'll talk a lot more about communication in the future. Homeostasis and control systems. So homeostasis, you should definitely know all of the vocab words related to homeostasis. So what is homeostasis? It's the maintenance of a stable internal environment, a constant internal environment. Homeostasis is the maintenance of a constant internal environment. The internal working conditions of the body are best suited to a constant internal operating range. How many chemical reactions take place in your body? A lot. And they need specific conditions in order for all of those reactions to take place. You need to maintain those conditions in order to have a functioning human being. Summarize the basic components of a cell. This should be mostly review from old classes for people. Please know all these terms. Please review all these terms. Be ready for probably 1 to 5 questions, a significant portion on this material. This section right here. Okay, so this top part is a lot of kind of review. Right here, once we get to cell membrane, this portion of the answer is new for most people in this class. You should know about the cell membrane. You should know that it's primarily made up of a phospholipid bilayer. You should know the functions of that membrane. Physical isolation, it separates outside from inside. It allows you to have a unique environment inside that membrane. Support structure, so embedded proteins and cholesterol help provide support and maintain that three-dimensional structure, form, and function. You probably talked about different cell shapes in anatomy class. Form and function matters at a cellular level. Semi-permeable membrane. You need to allow things to move in and out of your cell in order to get rid of waste products and bring in new usable materials. Communication, cells need to be able to coordinate with other cells. Separation of charge, we haven't talked about too much now. We did talk some about the sodium-potassium pump. So sodium-potassium pump, separates charges. It causes there to be more positive charges outside the cell than inside the cell. We'll talk a lot about this for test 2 with regards to nerve cells. Right here's phospholipid bilayer and cholesterol. You should know the different parts of the cell membrane, the difference between integral proteins and peripheral proteins, glycoproteins, glycolipids. You can just break down the words and know what those are. This chart right here, there will be a few questions directly from this chart. Again, this should be reviewed for most people, so be ready for those hopefully easy points. Outline the methods of both passive and active transport in which substances can cross the cell membrane. Make sure to state what the actual mechanisms are and whether or not energy is being used. This is a big right here. You should know these different mechanisms. You should be able to describe what's happening, the mechanisms for each of these, and know about energy usage. Both of those are going to be really important. There will be test questions on this. Be sure to include any pertinent information regarding the specifics of each mechanism. So first off, you want to kind of break this into categories when we're talking about transport across a membrane. Is it passive or is it active? Passive relies on the kinetic energy already within the system. It relies on concentration gradients. With passive transport, the energy required is already set up in a concentration gradient. Things move from high concentration to low concentration, high concentration to low concentration. Now there's different mechanisms. We'll get into those in a moment, but everything that's passive transport will have those characteristics. Active transport relies on the input of energy from ATP. With active, the energy is from ATP. It could be used directly or indirectly depending on the system in question. And there's a few types of active transport. So uniport moves only one type of molecule in one direction. Uni means one molecule, one type of molecule, one direction. Co-transport or co-transporter moves multiple types of molecules at the same time. What's an example of a co-transporter that we've talked about in class? Sodium-potassium pump. It moves two types of molecules at the same time. It moves sodium and potassium. It can be in both directions or only in one direction. So which one of these, how would you describe the sodium-potassium pump using either symporter or symport or antiport or antiporter? If you need to go back and review those slides, please do. So they move it in opposite directions. So sodium gets pumped out and potassium gets pumped in. So that's going to be an antiport or antiporter. Passive transport mechanism. So simple diffusion. So simple diffusion is just going from high concentration to low concentration. We're actually not talking about a membrane with simple diffusion. This is like moving from one part of a cell to another part of a cell, one part of interstitial fluid to another part of the interstitial fluid. It's just like substances moving from areas of high concentration to low concentration. It's like how you put a sugar cube in your coffee and those sugar molecules dissolve and just kind of spread throughout the whole coffee. Diffusion through a membrane, so this is where we have diffusion, like with A here, but it's across the membrane. Movement of like substances through a semi-permeable membrane from areas of high concentration to low concentration. Determined by the factors in Fick's law. No Fick's law. There will be questions about Fick's law. We have another question coming up about Fick's law where we're really going to get into it, so stay tuned for that. Facilitated diffusion through channels. Movement of like substances through protein channels in the membrane from an area of high concentration to low concentration. So these are the ones where, through channels. So depends on the number of channels present and the number of those channels that are open. So these are like the tunnels where molecules can flow through the tunnel, through the membrane. Some of them have gates, some of them have 2 gates, some of them have 1. Some of them are always open and have no gates. Unlike protein transporters, channels never saturate in the human system. With transporters are a little different. It's movement of like substances using a protein transporter from an area of high concentration to low concentration. So this is the one where you have a protein and glucose binds to the protein changes shape, and that shape change allows glucose to move through the membrane to the inside. Glucose then unbinds from the protein, and the protein changes shape back to its original state, and it can do it again. So there's never a constant direct tunnel, never a constant connection, never a constant open tube from the outside to the inside. It's due to a protein changing its shape and transporting something inward. Through channels, there is a constant, a direct channel through. And osmosis. Osmosis is water. What can do osmosis? Osmosis is the movement of water. Osmosis is the movement of water. I'm repeating this because some students have had trouble with that in the past. Osmosis is the movement of water across a semi-permeable membrane in response to a concentration gradient. Water moves from high concentrations of solutes to low concentrations. Water moves from higher water concentrations to lower water concentrations. That means water moves from lower solute to higher solute concentration. Water moves to dilute. The solution to pollution is dilution. Water moves towards areas of higher dissolved particles. We'll have another group problem on osmosis also coming up shortly. There are 4 basic types of active transport. What is active transport? Active transport uses ATP. With primary active transport, ATP is directly used as the fuel to move things across the membrane. Glucose binds to the transporter, ATP binds to the transporter, boom, it moves it across. Secondary active is it uses the potential energy stored in the concentration gradient that was established by primary active transport. Uses potential energy stored in a concentration gradient that was established by primary active transport. This is often considered to be indirect use of energy since the ATP that fuels this process is already used in primary active transport. Endocytosis is the process of capturing a substance or particle outside the cell and engulfing it within the cell membrane and bringing it into the cell. This requires ATP but does not use it directly. There are 3 types. Know the different types: phagocytosis, pinocytosis, and receptor-mediated endocytosis. In phagocytosis, the cell membrane engulfs the substance to be absorbed and then buds off to form a vesicle. It's non-specific. Pinocytosis is similar, but engulfs a drop of fluid. Non-specific. With receptor-mediated endocytosis, it's extremely selective. It's a substance binds a receptor, and that causes a reaction where the cell membrane kind of goes inward and brings that particle into the cell. It's very specific. Exocytosis is the process of a vessels fusing with the plasma membrane and releasing their contents to the outside of the cell. Right, here's a summary chart. You should be familiar with the summary chart. Distinguish between osmosis, osmotic pressure, osmolarity, osmicity, and tonicity. Then complete the following table for each row and table. Assume that the first row are perfect. Practice these problems. Practice these problems. There will be questions on the test on this material. So osmosis. Osmosis is the net movement of water across a semi-permeable membrane in response to concentration gradient. It's the movement of water. Osmotic pressure is the pressure due to the number of particles dissolved in a solution, regardless of size. So it refers to the force that is pushing water towards or pulling water towards the area of higher concentration. The pressure, the force of water being pushed in that direction. Osmolarity is the total number of particles in a solution, irrespective of size and charge. It's a measure of osmotic pressure. Osmicity is the initial difference in osmotic pressure determined by the total particles in the solution. Whether the particles move across a membrane does not matter to osmicity, just look at the initial conditions. By convention, it's always compared to the cell, the inside or outside relative to inside. So isoosmotic, the initial condition shows that no net difference in osmolarity. Hypoosmotic, the initial condition shows a lower concentration of particles outside the cell. So outside relative to inside. There's fewer outside, it's hypo. With hyper, there's more outside than inside. Tonicity describes what happens to the cell when it's placed in that solution based on the net movement of water. It really describes what happens to the cell, but is in reference to the solution. So isotonic, same inside, same outside. Hypotonic, the solution has less particles. Hypo, water moves towards the area of higher particles, so water moves into the cell and the cell swells. Hypertonic, more particles outside, so water moves to the solution out of the cell shrinks or crenates. This is super important right here. Make sure you're working in the proper units. Everything must be converted to milliosmoles so the comparisons can be made. You should do this before attempting to solve the problem so that the first thing you can do in each problem is perfect. Know how to calculate milliosmoles. I do not provide an equation sheet for the lecture exam like I did for the lab exam. Milliosmoles is equal to millimoles times the number of dissociable particles. 3 terms in this equation. Commit it to memory. You should know the number of dissociable particles when put in water for the following substances. There will be questions on this topic on the exam. You only need to know these 5 materials, these 5 molecules. All of the questions I will ask will be about these 5 molecules. You need to know the number of dissociable particles for each. To determine osmicity, you look at the initial conditions. Okay, so this is just going to walk you through all the basic steps. And then there's 9 practice problems. 9 practice problems. All right, I'm going to walk you through a couple. I'm going to point some things out. 280 milliosmoles of glucose is the solution. Step 1 is to make sure we have the right units. Make sure we're comparing apples to apples and oranges to oranges. At the starting point for this question, you are in milliosmoles. You do not need to use the equation to find out milliosmoles because we are already in milliosmoles. Compare that to question 2. So question 2 says 360 millimolars of urea in the solution. We have 280 milliosmoles in the red blood cell. So we're comparing apples to oranges here. We're comparing millimoles to milliosmoles. You need to convert millimolar to milliosmoles for question 2. How do you do that? 360 millimolar times 1 dissociable particle for urea, 360 milliosmoles. In question 1, we have 280 milliosmoles, 280 milliosmoles. We're comparing apples to apples. We're comparing milliosmoles to milliosmoles. There's no need to change the milliosmoles here into milliosmoles because it's already milliosmoles. Perfect. Down here, 280 milliosmoles urea. No need to change into milliosmoles because it's already there. Right here, 140 milliosmoles calcium chloride. Compare that to 280 milliosmoles. That's apples and oranges. We need to convert the millimoles into milliosmoles. So how do we do that? 140 millimolar times 3 dissociable particles. Perfect. The other thing, the other main area that students have trouble with is when dealing with urea. Urea is the one particle that we'll use that can go through the cell membrane. It's the one particle we'll use and go through the cell membrane. So you calculate your initial osmosity, then your final tonicity, your final osmotic pressure, you need to have urea move through the cell membrane. So how do you do that? You take half of the milliosmoles of urea and you add it to the 280 of the red blood cell. You add it to the 280 of the red blood cell. So you have 360 milliosmoles for your initial osmosity, 360 milliosmoles of urea. Half of that is going to be 180. So your solution is going to be left with 180 osmotic pressure milliosmoles. But you also need to add 180 to the red blood cell before you compare the two. So 280 plus 180 is going to be the final for inside the cell. 180 is going to be the final outside the cell. A lot of students forget that you need to add that 180 to the count for the red blood cell. Add it to the count for the red blood cell. All right, make sure you practice those. Fick's law. So Fick's law of diffusion. Explain what will happen to the diffusion rate in both quantitative and qualitative. So qualitative is it goes up, it doubles. The rate goes to half. You know, it's just kind of like it goes up, it goes down. Quantitative is going to like put numbers to it. It's going to say 3 times, 1/2, 0.3. Terms with each of the following examples. In each example, assume that the only parameter that changes is the one given in the problem. So Fick's law of diffusion is concerned with the diffusion of substances directly across a cell membrane. Thus only applies to molecules that can pass directly across the cell membrane, which are typically smaller nonpolar molecules. There's 4 variables. So net flux is equal to available surface area concentration times concentration gradient divided by membrane resistance and membrane thickness. Now, you should understand that surface area and concentration gradient are directly related. You increase these variables, you increase net flux. Membrane resistance and membrane thickness are inversely related. If you increase resistance, you decrease flux. All right, so when doing these problems, what I recommend is you write out the equation. You write out net flux is equal to SA, concentration, surface area times concentration gradient over membrane resistance times membrane thickness. And then you put a 1 in front of each of those variables. Then read the question. An experimental drug is given that increases membrane thickness to membrane resistance to oxygen of mitochondria by 4-fold. What happens to the diffusion rate of oxygen in the mitochondria? So increase by 4. So you've got imaginary 1s in front of your 4 variables. Put a 4 in front of membrane resistance. So right here in front of membrane resistance, we're going to put a 4. And now calculate it. So we've got 1 times 1 in the top divided by 4 times 1. So you've got 1 over 4. 1 over 4. We know that the membrane resistance is inversely proportional. Blah, 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 blah. Therefore, we can say that qualitatively, there was a decrease in net flux, while quantitatively, the change was 1/4. So 1/4 is going to be the final flux. An extremely rare genetic disease triples the thickness. Triples the thickness. You're going to put a 3 in front of thickness. Put a 3 in front of thickness. Cell membrane. Oh, what happens to fusion rate of sodium? Oh, sodium doesn't move through. Okay, so remember that Fick's law of diffusion only applies to substances that can directly cross the membrane. This means that molecules that are small and nonpolar, since sodium is polar molecule, does not directly move across the cell membrane. Effects would be 0 because sodium doesn't move across. If we used urea in this example, some sort of small nonpolar molecule that can move across, you put a 3 in front of thickness. This one, hyperbaric oxygen therapy, is given to the patient that doubles the concentration of oxygen. So you're going to put a 2 in front of concentration gradient. It's going to double the rate, and so on. All right. If you have any questions, please let me know. There's a few more that you can practice with here. But be ready for questions like that. Outline the general principles involved in enzymes and their activities due to principle. Perfect. So this material is going to be from the enzymes and energy lecture. Read over this. Be familiar with this. Bonus question right here. So explain the types of channels that can be found in cell membranes, including both the general types of channels and specific types of channels of each. So we did talk about this a little bit when we talked about passive movement across the cell membrane. You have channels that are gated and non-gated. And some of the gated channels, there's different things that open up those gates. It could be chemically gated, it could be voltage, mechanical, time, light, temperature. Some channels have 2 gates, some channels have 1 gate. All right, good luck studying, everybody. If you have any questions, please reach out to me.